Good evening, everyone, and welcome to the Maryland Entomological Society April 17th, 2026 regular meeting. Our speaker tonight is Dr. Christopher Oufiero, Professor of Biological Sciences at Towson University. The title of tonight's talk, A Mantis in Leaf Clothing. Does camouflage evolution shape the diversity of predation among Mantodea? Our speaker, Dr. Christopher Oufiero, received a bachelor's in biology from Montclair State University, New Jersey. After working as a zookeeper, he completed a master's at Indiana State, where his thesis focused on the growth and development of the eastern fence, and a PhD at the University of California, Riverside, where he studied the locomotor costs associated with the sexually selected sword found in swordtail fishes. Was it electric fish? I hope that it says fence on here, and I just read it through, but
Fence lizards.
Yeah,
fence
fences.
lizards. Okay, that makes sense. It was a typo that the editor left out lizards on the flyer. Okay, sorry about that. Since 2012, he's been at Towson University, where his lab has been focused on studying the evolutionary physiology and functional morphology of praying mantises. All praying mantises use their multi-segmented raptorial forelegs for prey capture and mandibles for ingestion. These feeding apparatuses are used across the diversity of camouflage strategies that evolved in the group, including flower, dead leaf, stick, and bark mimics, to name a few. The evolution of camouflage strategies has been hypothesized to result in ecomorphs, where species evolve specific morphologies in response to ecological pressures to function in their environment. The goal of this research is to examine the role of camouflage evolution on prey capture and ingestion among habit-mimicking praying mantises in the context of body size and phylogeny, to determine if there is evidence of ecomorphs. The results suggest minimal influence of camouflage strategy on prey capture and ingestion. However, they highlight the versatility of praying mantis predation, which may have allowed them to diversify in camouflage strategies. Okay, so without further ado, I will let Dr. Oufiero take over the screen
Yes, thank
and
you.
share his PowerPoint with us. Thank you so much for being with us tonight. We greatly appreciate this.
All right, let me get can everybody see that, hopefully?
Yeah.
All right, well, thank you for that nice introduction. Thanks for having me tonight. Excited to sort of talk about this. This is the work of the lab over the past several years. And so with that, there we go. I like to start with acknowledgments. This work really is the result of a lot of student work, so undergraduate and master's students that have been in my lab over the years. And so I'm not going to name everyone right now because I'll be highlighting some of their individual projects throughout the talk. And just want to acknowledge the Fisher College of Science and Math for funding throughout my time here at Towson. So to start, sort of what I'm interested in, my lab is interested in, is I'm really interested in the evolution of physiological diversity, and I've really focused on movement in animals. And so these videos, and I'm going to have a lot of videos in this talk today. This is sort of the focus of a lot of what I do. But I studied I focus on movement for several reasons. And one is that it's an integrative and complex trait, meaning that like movement itself is the result of many underlying sort of biological functions. So muscular physiology and sensory physiology and the skeletal systems or exoskeletal systems. And it's related to the morphology of the organism and it can be affected by the ecology. And more importantly, is movements often related to fitness or important to fitness. As you can see in a lot of these videos, it really determines whether you can grab prey or potentially get away from being prey among organisms. And so all of these videos are from a class I teach here at Towson, which has been changed to the Animals in Motion Cure. This has been done over the past several years. And throughout my lab, we really look at two sort of contexts. We look at the within and among individual variation in movement, and then do evolutionary comparisons taking into account the phylogenetic relationships among those organisms. So I've been focusing the past several years here at Towson on predation in mantises, and there's a couple of reasons for that. One is that if you look at the earlier models of foraging ecology, mantises were actually one of the ones used in those models to sort of model optimal foraging theory. The other is that they capture and consume evasive and live prey. And so in the lab, we like to highlight that mantises are capturing live evasive prey without the use of anything like a venom or a web or any elastic mechanism like tongues that shoot out. This is all the result of sort of direct muscle activation of the forelegs. And they're doing it with these two multi-segmented forelegs that I'll talk more about, that they sort of throw out into this three-dimensional space to capture prey. So we've really been focusing on this predation among mantises for these reasons. Mantises in general, there's about 2,500 plus species of them. They are ecologically and morphologically diverse. You can see some sample images here. One important component of their predation that we don't study, but a lot of great research has been focused on is their stereopsis, or their ability to perceive depth, which makes a lot of sense if you're throwing two forelegs out into the environment to capture prey. Is probably beneficial to be able to distinguish depth when you're doing that. They're all predators. They all use their raptorial forelegs for prey capture. And what's been the focus of our lab recently is they have these diverse camouflage strategies. And so among mantises, they have some of the most diverse camouflage strategies in the animal kingdom. And these are just some examples here of ones we've had in the lab. So a bark mimicking, a dead leaf mimicking, flower mimicking, stick mimicking. And then there's a lot that are just big and green, but nature is mostly green. And so whether or not they're camouflage or generalist still remains to be determined. Some of this camouflage you can see that's not only coloration, but some like the flower mimics and dead leaf mimics have these lobes on the legs to sort of give them a bit more of appearance like the habitat they're mimicking. Some like the dead leaves have this widened pronotum that's again trying to blend into that dead leaf environment a little bit more. The orchid mantis, they have a lot of sort of hypermimicry. They not only look like flowers, but they emit pheromones to smell like flowers and attract predators. They give off UV coloration to attract pollinators as well. And some of this camouflage may come down to posture. So something like this African twig mantis holds its forelegs out to sort of give that twig-like appearance, whereas some bark mimics really hold themselves close to the bark to sort of flatten and blend in a little bit more. Camouflage has been studied a lot in the animal kingdom, and it's mostly been studied from a prey's perspective. And this is because it allows an animal or organism to remain undetected or seen, so hopefully not being predated upon. And most of the research has focused on how it works, how the sort of sender and receiver are sort of integrating and receiving and sending these signals. But prey aren't the only ones that have camouflage. We do see it among predators, which hasn't been studied as extensively. And so these are just some examples here. This is a frogfish that tends to blend into sort of the rocky and coral environment. Some large carnivore predators, although they may look orange to us, their coloration and patterning does allow them to blend in. Some of their prey don't have the ability to see red, and so they kind of just blend right in with that environment. And this is another example of a fish predator. These are stonefish that again blend into that stone environment. And you most often see camouflage involving in predators that are ambush or sit and wait. So they sit, they remain still, they remain undetected and unseen. And so this camouflage strategy may allow the prey to get closer and it may increase their chances of success. And recently, camouflage has been examined in stick and leaf insects, another great group that has lots of camouflage strategies. And this paper has shown that there is sort of these ecomorphs that have evolved. And so disparate lineages evolving similar morphologies to sort of function in that environment. And so we've really been interested in camouflage evolution in mantises because of their diversity and their sort of ambush, sit and wait predation. strategies with this idea, like this quote from a review on camouflage and predators, the idea that individuals that differ in their appearance may also choose to live in different microhabitats. So they might be encountering different types of prey. They might be functioning differently in these different types of habitats that they're mimicking. So we're interested in sort of, does camouflage evolution result in ecomorphs? Do we see any functional or morphological differences among these habitat mimics? Or is camouflage more decorative and not really introducing the form-function relationships of predation? So our ultimate goal is to understand how camouflage evolution relates to diversity and predation among mantises. So when we think about predation, there's several stages that occur. The first is an encounter, which we simulate in the lab, and that's followed by detection, where some aspect of the prey is cueing in on the predator's sort of sensory system, so it detects it and cues in on it. That detection, as it sort of hits the right note, is going to elicit an attack that you'll see here in this video. But one thing to note, and I'll mention this later, is attacks aren't always perfect. Predators do miss. And so they might have to go back to this detection phase, and something has to sort of trigger that again. And there's been lots of great work in mantises on this sort of detection phase, what sort of elicits the strike. Once they initiate another attack, and if they're successful, they capture the prey. They then have to subdue it, which then leads to the next stage in mantises, which is the ingestion. And again, mantises consume prey alive, so they're using the force in their forelegs to grip a living prey, and their mandibles to shear off pieces of that prey. After the ingestion phase, you can see in this video, the abdomen will swell as it eats the cockroach. They'll digest that prey item, and it could take up to 96 hours for them to fully digest it. So among this predation strategy, what we've really been focused on in the lab is this attack prey capture phase and the ingestion phase of predation.
Open the door.
So for prey capture in mantises, you'll see a video down here, sort of that'll loop through. So prey capture mantises, they're using various cues of the prey that's sort of eliciting that detection and that attack itself. So movement's really important, and the distance of the prey is really important. And again, they're using the coordinated movement of these two multi-segmented raptorial forelegs. So they have this elongated coxa, a trochanter femur with a series of spines, and a tibia with a series of spines. They also have a tarsus, but that tarsus is folded back during prey capture. So the goal is to get the prey in this sort of prey capture space in between the tibial and femoral spines. With praying mantis prey capture, there's two main stages that have been identified. There's some great earlier research that have characterized these stages. And the first you can see is this approach, which is the abduction and extension of the tibia. And that's followed by the sweep, which is the sort of simultaneous extension of the femur trochanter and the flexion of the tibia, trapping that prey item between those femoral and tibial spines. We'll show that these prey capture responses are variable, sometimes in response to prey capture. And as I showed already, they're not always perfect. They do miss. So how do we capture prey capture in the lab? The way we do this is we use high-speed cameras. We film at a frame rate of 1,000 frames per second. Most of the videos today will be played back at 30 frames per second. So here's our current setup, and we're doing this in 3D. So we have two high-speed cameras, one capturing a lateral view and one capturing the ventral view. We put the mantises upside down on this platform over here. They tend to prefer to feed upside down, so this is their more preferred position. So there is a mantis actually in this image here. We have a series of lights at a high frame rate. You need a lot of lights, so we have this bright LED light and a couple of infrared lights that the camera sensors can pick up on. We then use a combination of machine learning and manual tracking. We track 10 points along the body and forelegs of the mantis, as well as the prey. And so this is showing a result of a machine learn model. What you'll see is it's not perfect. So there is imperfection of this as well. So some of the points jump around. So we have to go in and manually sort of clean that up. And from this, we obtain a suite of kinematic and behavioral variables after we track the prey capture strike. And so this is another example. This is Staglomantis limbata, a prey capture attempt. And this is a resulting sort of plot we get showing some of these kinematics. This top plot here is showing the angle of each joint. On all of these, time is going to be on our x-axis. The coxas in green, trochanter femur in blue, front tibia in purple, and back tibia in gray. And you see this sort of series of events that's pretty standardized across mantises, where you have that opening of the coxa and extension of the tibia, and then you have this quick flexion or extension of the femur and flexion of the tibia. On these plots, just showing this is the time of the approach. So we're getting that from the videos. This red dashed line is when the prey is entering that prey capture space, so really when the mantis is capturing the prey. We also get the distance of each of those joints and the linear velocity as well. So we pull a lot of variables from these videos, and I'll show some here. So for each joint, we're getting the maximum angle that's achieved during the strike. We also get the angular velocity, so it's a rotational velocity of each of those joints. We also get the linear displacement, so like this trochanter femur, it's linear displacement throughout the strike as well as the linear velocity. We've been filming in 3D, which has been great because now we can get the lateral displacement of each of these points along the mantis, so how wide they're opening up their forelegs. We also get their body displacement and velocity. Some mantises you'll see use a lot of lunge movement towards the prey to sort of close that predator-prey gap. And then we also get predator-prey distance and angle, which isn't shown here in this image. So we get a suite of these kinematic variables throughout these prey capture strikes. So some early research in the lab, just to sort of highlight what we've done, looked at within species or intraspecific variation in prey capture. So sort of the first studies we looked at, we were using the ghost mantis, and this was on subadults, and we were really just seeing how variable the strikes were. Are they sort of little robots doing the same thing over and over, or are they variable in sort of the kinematics and behaviors during a prey capture attempt? So what you see here is this is each joint, the coxa, femur, and tibia, and these are the angles at the end of the approach as squares and their maximums in circles. And we saw that the angles at the end of the approach tended to be more variable than the maximums, which makes sense because they are an organism and have some constraints on how much they can sort of extend their forelegs. We also saw that there was a relationship between how far away the prey were, the predator-prey distance, and how much of their body, the percent lunge that they use. So the further away prey were, the more they're lunging to the prey to close that predator-prey gap. We then expanded on this and tracked ghost mantises throughout ontogeny through development and compared these kinematics with body size. And so on the x-axis, we have body size for both of these. And then this first plot is the linear velocity of the forelegs and then the angular velocities. And what we found is that the linear velocities are increasing as mantises grow, which makes sense because their forelegs increase as well. But we saw minimal changes in the angular velocities across ontogeny. So they're not changing how fast they're rotating each of those segments, but simply by growing larger, that's resulting in some faster linear velocities as they get bigger. More recently, we've started to look at whether or not or what's predicting success in prey capture attempts. And so this was a data set on one species of mantises, all adult females, where I saved both successful and unsuccessful prey capture attempts, trying to get an idea of why do they fail, even under laboratory conditions where everything seems perfect and we're kind of controlling the prey. And what we found is that capture success really is due to how far away the prey are, as well as the speed of the attempt. So you can see here on this PC1 from a principal component analysis, so we take all of these kinematic variables and we put it in a principal component to sort of describe these functional spaces. The farther away the prey were, the more expansive and fast the prey capture attempts were, but that resulted in a decreased probability of successfully capturing the prey. Versus when the prey were closer, they had these reduced, slower attempts at prey capture. And this hints at potentially trade-offs between sort of the speed and accuracy. You may be able to be fast, but that may come at a cost to how accurate you can be in capturing your prey. More recently, this is unpublished work. This is from MK. She was an undergrad in the lab. and just defended her thesis 2 weeks ago. And so this was some of her thesis work, where she was looking at the relationships between prey size and prey capture kinematics. And so we started to see some differences with distances, and there's been some studies on movement of prey, but we wanted to know, are they able to sense the size of the prey and adjust their prey capture strikes? So I'll show this video here. And again, these videos are the ones we use in the research. This is a orchid mantis showing both views. That's on a medium-sized prey. And now this is a larger prey. We used cockroaches because we can get them in variable sizes. And she did this in adult females across 2 species, an orchid mantis, which really has this sort of hyper mimicry style, versus the giant Asian mantis, which is just a more, I don't want to say general, but big green mantis, what we kind of think of as a general mantis. So she measured the prey capture strikes in these mantises. She has over 200 videos that she analyzed, and she put both species in a principal component analysis again. And I'll be showing several of these throughout the talk tonight. And so just to kind of orient you to these, each point on this space is a prey capture attempt. And what these arrows represent, they're vectors. So they show both the sort of direction and magnitude of the contribution of each of our kinematic variables. So in this case here, our giant Asian mantises, the hierogula, come out on this side of the PC and are really characterized by a longer prey capture time and more lateral displacement of those forelegs. They're opening up more in this position. On the opposite side of this, we have our orchid mantises, and you can see they're really characterized by faster strikes, so higher velocities of the femur, the tibia, and the coxa. The point size here represents the relative prey size. So orchid mantises are a little bit smaller, so they tend to have a little bit larger relative prey size compared to the hierogula that we used in this. But our question was whether or not prey size is affecting it. And this is some preliminary analysis. We're still working on adjusting these, but it does look like that as relative prey size increases, there's increased lateral displacement in both species. So a potential strategy, they're sort of able to sense that the prey is larger and they're adjusting those prey capture attempts accordingly. Again, we're still working on analyzing this. So this is just sort of some preliminary analyses. So all of those previous studies have really shown that prey capture varies and it can be modulated in response to prey, both size and distance. But going back to our original question of how camouflage relates to this, this was really the work of master student Lohit, who finished his MS degree here at Towson in 2024. He's currently a PhD student up at the American Museum of Natural History with Dr. Jessica Ware, continuing to study the evolutionary relationships of mantises. But he, for his master's thesis, was really looking at this question of how camouflage evolution affects prey capture. So he measured 10 species of mantises, you could see here in this phylogeny, from 4 camouflage strategies. Flowers represented by this purplish colors throughout, dead leaves represented by squares and this orangish reddish color throughout, stick mimics, triangles represented by brown colors, and what we call generalists represented by just sort of greenish colors. He had 3 to 5 adult females for each species. These were obtained from the hobby industry, some of them raised up here in the labs at Towson. He did 3 to 5 prey captures per individual. We fed them a standard prey, so we wanted to standardize the prey to really see the differences among the species. So we used blue bottle flies. And at the end, he had more than 230 prey capture attempts among these 10 species. And so we also looked at the morphology. So I'll show you that first from the videos. We looked at the morphology of the raptorial forelegs, so the length of the tibia, the femur trochanter, the coxa, and just the body size length based upon our points. And it doesn't come out that it looks like camouflage is really affecting this raptorial foreleg morphology. So on this is a principal component analysis. On PC1, it explains 68% of the variation. Longer coxa and femur and metazone, bigger mantises on this side. Shorter coxa and femur and metazone, smaller mantises on this side. PC2 shows a sort of trade-off between tibia size, so shorter tibial species on top and longer tibial species in the negative. And in all of these PC plots that I'll be showing, these smaller transparent points are the individual means of a species. These larger, more not transparent points, these are the species means. And then the black lines represent the phylogenetic relationships of the species. So for this part of the study, it looked like there was no support of ecomorphs of the raptorial forelegs among the species that we studied. Moving on to the kinematics, this was a little complicated, so I broke it down to sort of step through it. We took the three fastest attempts per individual and ran a principal component on that. This is to ensure that these are really the motivated strikes. We can't tell a mantis to strike as fast as you can, so we take the three fastest ones as an indication that these are the really motivated prey capture attempts. And so among those 230 prey capture attempts that we use, this is how the kinematics are sort of contributing to PC1 and 2. So just to sort of orient us, on this side here, this is all angular and linear velocities. So tibia angular velocity and femur angular and linear velocity. And that's sort of trading off against our lateral displacements of those forelegs. It's also trading off with a sweep time. So this is a longer sweep time on this axis. So PC1 is really this faster versus more lateral displacement and longer times during a prey capture. PC2 is a trade-off between how much velocity of the body and the coxa linear velocity they're using versus how long that approach is, the sort of setup to the prey capture attempt. So if a prey capture falls in any one of these spaces close to these, like if it falls over here, it's saying that it had a fast coxa angular velocity, a fast femur velocity. So looking at all of the individual prey capture attempts, we can see that sort of functional space among praying mantises. And a couple of things to point out, you can start to see some groupings among species within these. So we then got means of individuals and species means, and we used a phylogenetic general linear mixed model. So what this does is basically taking into account both the phylogenetic relationships, because similar species might function the same, but it also takes into account the within species variation, the fact that not every individual within a species is functioning the same. And you can see that on these smaller transparent points. So what did we find? Along PC1, this sort of fast versus lateral displacement, there was no effect of camouflage, and really this seemed to be separating out by phylogenetic relatedness. So on this axis here on PC1, the lower values, these are all Hymenopodidae, so the flower mimics and the one deadleaf mimic nested within that family. When we looked at PC2, we did start to see an effect of camouflage. Mainly, our two deadleaf mimics seem to be taking a longer time in that setup during that approach phase of the strike, and that trading off with how fast they're moving their bodies. So they're taking longer for the setup and not moving their body as fast as some of these other species. To sort of pull out some of these kinematic variables to make it a little bit easier to digest, this is just showing here's our phylogenetic relationships and our species that we use. And we have femur angular velocity, tibia angular velocity, and body displacement. So you can see our Hymenopodidae, our flower mimics, tend to be faster than some of the other species, but aren't using as much of their body. And you can see that especially with the deadleafs, don't really move their body that much at all. Looking at some of the PC2 traits that really were significant on the axis, you can see that some of the deadleafs have a longer approach time and less coxa linear velocity and body velocity. We also looked at the third principal component axis. So these plots are the same, except for now on the Y, we have the third PCA. And this is really described by, if you look over here, species that have a higher coxa starting angle. So they're kind of, if you can see me, starting with their coxa at a position up compared to the typical sort of praying position of praying mantises. And there was no effect of camouflage on PC3, but this is something we've highlighted because it does seem to be a unique trait and potential behavioral difference among mantises. So our orchid mantis is sort of exemplary of this, and we have another flower mimic that also exhibits this. and one stick mimic that's sort of converging on what we're calling an anterograde prey capture position, compared to a basigrade, which is keeping that coxa at a lower angle at the start of a prey capture attempt. So I'm going to let these videos play, and what you'll see is all example of all the species we studied, so all 10, generalists up in this corner, flower mimics next to them, dead leaf stem in this corner, and stick mimics. And you'll notice some differences just by watching them. Timing of events, you'll see the flowers are very fast. You'll see the stick mimics tend to use more of their body. So I'll let these play as I sort of summarize our prey capture results. We found that it does vary within and among species, and within species, it's really modulated in relation to prey distance and size. Among species, there seems to be a strong evolutionary component, so similar species do function similarly, with some relations to camouflage, but not sort of strong relations. So we really haven't found strong support for ecomorphs within praying mantises in relation to camouflage. So after prey capture, a mantis subdues the prey and then ingests it alive, like I mentioned, using the shearing action of their mandibles. So this next part is focused on that ingestion phase. And ingestion is interesting because it's part of a predator's functional response. What's been modeled in ecological models is handling time. And they consume the prey alive, they use their forelegs to grip it. An increase in ingestion may in time, so how long they're sort of processing that prey might make the mantis more susceptible to predation, and it might mean less time for them to forage another prey item. So the goals of this part of the study were to again look at the effects of camouflage, but also look at the allometric variation, how ingestion relates to size. And we were kind of interested in how the energy intake, this ingestion, relates to the energy use metabolic rate. So we didn't measure metabolic rate, but we compared our results to previous studies of metabolic rate. For this, we use time-lapse cameras. So instead of slowing things down, we actually have to speed things up because while prey capture lasts the third of the time it takes to blink our eye, so 50 to 100 milliseconds, ingestion can take anywhere from 30 seconds to up to an hour, depending upon the predator-prey size relationships. So this work was really the result of two undergrads at the time, Lizzie McMillan and Marley Wood. Lizzie has graduated and is now currently a middle school teacher in the area. Marley is currently a first-year master's student in the lab. So they measured 1 to 8 adult females. They did 3 to 5 trials per female with time-lapse. We increased our sample to 14 species, so we have some additional flower mimics in the data set and some additional stick mimics in the data set. So there's a total of about 66 individuals. They did 324 trials, filmed them at 1 to 2 frames per second. And again, we use the standard prey for this, looking at the ingestion time, standardizing that prey. We use a combination of phylogenetic general linear mixed models like we did before. So taking into account those evolutionary relationships, but also with the within species variation. And then phylogenetic general least squares, which is just on species means, just for comparisons. Colors are the same and shapes are the same. So flowers are purplish diamonds, dead leaves are orangish boxes, sticks are brownish triangles, and generalists are greenish circles. So what did we find? So what this plot's showing is predator mass on the x-axis, and we log 10 transform the data for two reasons. One, we want it to be comparable to studies of metabolic rate and other studies of handling time. And so log 10ing is sort of standardized because a lot of these relationships are a power function, so this makes it linear. And the second is we had a pretty big size distribution, and so this helps sort of visualize it and make the data more normally distributed. But what we found is that, and then on the y-axis here is just the ingestion time in seconds. So higher the ingestion time, the longer they take to consume that standard prey. And we found that as species size increases, that the time to ingest decreases, which sort of makes sense. The bigger you are, the faster you can eat. We found a minimal effect of camouflage. Our dead leaves fell outside our confidence intervals, these dotted lines here, but it wasn't significant. So they tended to eat a little bit more slowly compared to other species, but not significantly so. And this is just showing the slope of the regression of negative 0.841. To sort of take into account the effect that we do have different size species, but we're feeding them all standard prey, we use a common metric in functional response models, which is sort of this ingestion time divided by prey mass. So trying to standardize that by the prey mass to compare to other studies. And we get a similar relationship. Bigger species, as size is increasing, there's a decreased time to ingest prey per mass. So basically, bigger species they're eating the prey faster than the smaller species. But what we did next was there was a large database available from a recent publication that has handling time among a whole bunch of species. And so on this plot, this is showing was the fitted handling time from this forage data set. So other insects are gray circles. There was some mollusks in there, reptiles, fish, mammals, and arachnids. We really wanted to compare as a predator, how does the ingestion scaling compared to other predators that are in this data set. So for any species or group that we had more than 10 species, we fitted a linear regression line. We then fit a linear regression to our data set like we did earlier. So our data points are here, and then the purplish lines are our mantises that we studied, and you can see they fall right within the grouping of others. What we found is that mantis scaling for ingestion is really similar to arachnids, which are this black line here, different than other insect predators, this gray line here. When we compared it to their metabolic scaling, we found that both mantises and arachnids had greater scaling for their metabolic rate or for their ingestion rate than their metabolic rate. So for their metabolic rate, the scaling, this is just some numbers, was about 0.74 for mantises, I think 0.6 for arachnids. Their ingestion was higher, 0.8 for mantises, and a little bit higher for arachnids. So what this means essentially is that they're ingesting, taking energy in at a faster rate for their body size than they're using it. And this could have several functions and reasons. One is that, well, when they eat, not all of that ingested food is processed into useful food. Some is lost to weight, so that greater ingestion might be to compensate for anything that's lost in the process of transforming those food items. So these videos will show some of our mantises and our setup that we used in this, and I'll have those play in, as we did before. But body size explains most of the variation in ingestion, and mantises are consuming at a faster rate than they're potentially using energy. And camouflage doesn't seem to have a strong effect on how fast they're consuming prey. It's really about their body size in this data set. So here's one of our deadleaf mimics. You can watch that play, and you can see these videos have a timestamp, which we use to get the ingestion time. So we could set them aside, let them eat the prey, and not be disturbed. We don't have to watch them. The time lapse really allows us to leave them undisturbed. And any pieces of prey that's left over, we would weigh that. So all the prey masses that you've seen are an actual ingested. It's not the starting size of the prey, it's what they actually consumed. So to summarize so far, in relation to camouflage and predation in mantises, it seems to have minimal effects on the diversification of prey capture and ingestion. But what I think is interesting is that because prey capture is flexible and versatile in mantises, it may have allowed them to sort of expand their ecological niches and these different camouflage strategies. In relation to ingestion, while most of it relates to body size, it may help us understand the impact different sized mantises have on an ecosystem. Here in Maryland alone, we have several introduced species, ones that have been here for a long time, like Towson sinensis, the Chinese mantis, and more recent ones like the Asian jumping mantis, which was only introduced a few years ago, and I've seen more and more recently. So understanding how big a mantis is might give us an idea of the impacts it has on the ecosystem. But going back to our original question of ecomorphs, are there actually ecomorphs among mantises? And if not, what might explain this morphological diversity among mantises? These are just some images I took when I did a study at the Philadelphia Academy of Sciences in their collection, highlighting some of this huge diversity in their raptorial forelegs, their bodies, their heads, all components of them. Well, the first is that we need to measure more morphological traits. We've really been focused on linear measures of the forelegs. That might not quite capture really the morphological diversity of mantises. We've done some preliminary work using elliptical Fourier analysis on the forelegs to really capture that uh, shape of them. And the study of stick and leaf insects really did more morphological traits, so included other aspects of the body, the wings, and the other legs. And so we've been doing some preliminary work in the lab on that to see if maybe including more morphological traits allows us to see relations to these camouflage strategies. Marley, who's now a master's student, her thesis is going to be focused on using photogrammetry to characterize the shape of the eyes among mantises. And you can see some of that diversity. You have these pointy shaped vertical eyes, like in the orchid mantis, these more rounded eyes, like in the stick mantis, these pointy to the side eyes, and just sort of regular eyes. It's a bad description, but we're going to work on better describing that. She's also hoping to characterize the shape variation among the mandibles and relate that to the ingestion as well. And so we'll be dissecting out the mandibles and characterizing the shape variation of those. So perhaps we've just been missing some of this because we haven't measured enough of the morphological traits. The other thing is that there may be other components of the ecology, such as the habitat, where our ecomorphs fall out. So a classic example of ecomorphs are the anole radiation in the Caribbean islands, and they're really ecomorphs based upon where they're found in the microenvironment. So whether they're on the top of the trees, whether they're on the twigs, whether they're on the trunk, whether they're in the bush or the grass. And so I have these examples here of different mantis species. This is considered a bark mantis. Here's a Carolina mantis that do have this bark coloration, a budwing mantis that has a bark coloration from Africa, and an African twig mantis that has this bark coloration. But they're not all considered bark. This is a twig and this is a bark. So it might be where in the canopy or where in the environment they're found, not just sort of these gross anthropomorphic classifications we're giving them. And related to that, the other thing we're hoping to do is not have our view on it, but use what's called k-mean cluster analysis to group mantises or group the morphology using like maximum likelihood and algorithms. So it sort of takes the human side out of it because we're often saying, well, it looks like a leaf, so it's probably a leaf. But I've always been surprised with the Tinodera sinensis we have here that we just consider them a generalist. But when my cure class went out to Irvine Nature Center this fall to collect, we find them in the tall meadows and the grass, and they're hard to find. So who's to say that they're not actually a grass mimic, not just a quote unquote generalist? So this type of analysis will allow algorithms to sort of group morphologically or functionally, taking out our sort of anthropomorphic biases. But what else might explain morphological diversity? One of the big black boxes of praying mantises in a lot of animals is their actual diet. And so for a lot of vertebrates, you can go to museum collections, you can open up the gut contents. If anybody's ever done a shark dissection, this is always a fun part. You can see shrimp in there, you can see fish in there, and you can identify what the predator ate. Well, because mantises are shearing the prey alive, all that's left are pieces of it, so it's hard to get an idea of their diet from their gut contents. But Ryan, who was an undergrad in the lab and then did his master's here, graduating last year, he undertook a project using DNA metabarcoding to try and classify the diet of Tinodera sinensis across 4 sites here in Maryland. And he looked at, I'll show the results here, Cromwell Valley, Lily Ponds, which is out in Western Maryland by where he grew up, Oregon Ridge, and then here on Towson's campus. And so what each of these plots are showing is for each of these populations, on the x-axis is the number of occurrences in the diet, and the y-axis is the species that he found using DNA metabarcoding. So he sequenced the cytochrome oxidase 1 gene, which is pretty common, and then used pipelines to identify the species that were in the diet from the gut contents. Some of the main contributors across all 4 sites were lanternflies, which is good because they're also an introduced and invasive species, paper wasps, and skipper butterflies. And so they tended to be the most common throughout all 4 sites. His original master's was looking at testing the hypothesis of an urban-rural gradient. We didn't necessarily find that, but some preliminary results suggest there may be an effect of artificial light at night affecting what they're eating, with those in areas with more artificial light at night, eating more nocturnal species than those that don't have that. So, sort of wrap up everything we've been doing. I kind of like these two quotes. I teach a grad class here and was reading papers on optimality and design of organisms, and they kind of summarize what I think is happening potentially in mantises. And so this first one's optimization theory has shown its limits by demonstrating that the more complex and multitasking a phenotype is, the more likely one is to find many alternative optimal designs. And this other more recent one, a successful biological design is therefore flexible and sufficient rather than optimal. So we potentially think that might be what's happening with the raptorial forelegs of mantises. You have two of them, each composed of three segments, and so you have a lot of degrees of freedom that can be adjusted to capture prey. And so it might just be this versatile system that has allowed mantises to diversify ecologically. And so going back to my title of a mantis in leaf clothing, camouflage may just be ornamental and not related to the variation in predation. That might be something else that we haven't yet looked at, like the diet of the mantises or the habitat that they're living in. But camouflage is just sort of this clothing that mantises can put on and still function pretty flexibly in the environment. Summarizing everything that I've sort of said already, prey capture, they're using various cues to adjust their prey capture attempt. The mechanics are similar, but the sort of resulting kinematics do vary across species. I didn't mention this, but there's actually minimal effect of size on the prey capture kinematics, unlike ingestion, where size is explaining most of that variation. This may be related to mandible variation, which Marley will be undertaking, and both may have allowed them to diversify. So our future is to examine these ecomorphs in a broader context, more morphological traits, additional ecological variables, maybe getting more into diet. I think this could be a key. It's hard to get that data, but it may inform us what is they're actually eating. And then looking at variation in prey capture success on different prey. Sort of key to all of prey capture is you need to be successful, and so maybe there's differences among these different camouflage strategies. So with that, I'm going to leave you. This is a video, a time lapse. There is a praying mantis here. This is on our Towson campus. I had Ryan test this out. We were trying to get an idea of can we film a mantis in the wild and look at some of these components of foraging or functional responses of a predator. So how often it's encountering a prey and captures it. And so if it lets me, I can click on it. I think, yeah, it'll play along. It's a long video, so I can. I'll end there, and thanks for listening, and take any questions if there's time. This mantis does eventually catch a skippered butterfly. It tries a lot, but it will eventually catch one towards the end.
Looks like there's something bigger flying around too, but I can't
Yeah,
make it
there might be. There's also students walking around in the background.
out. Now, there's some other larger butterfly, it looks like flying about there.
This was about 2 hours of video that he took. I cropped it out, but he is in the background just sitting watching.
Mm.
But while this is playing, I'd be happy to entertain any questions if anybody has any.
Yeah, well, to what I've seen of these mantises, they seem to prey on just about anything, including bumblebees and other types of Hymenoptera. And I guess really whatever comes close to them is fair game, isn't it?
That's what we think. They're just really at least for what we see, especially the ones around here, they're opportunistic. So this is a
Mhm.
female. She just, perched on this plant, this flower that pollinators are coming to, and there she finally got
Oh
one.
yeah, yeah.
Um, and so she just waits and tries and misses and tries again. But think they're really opportunistic predators.
The skippers are pretty fast, so Mantis has got to be pretty quick too.
I don't know how to moderate this on Zoom. It's been a while since I've been on Zoom, but if people have questions, just ask.
Yep. Anybody with a question, please.
Oh, I have a question.
Yeah.
So I know you were saying that the mantises, like they rip apart their food, so you guys can't really test their like insides, like their digestion, like what they eat. But I was wondering, is there a test that will maybe tell you what kind of bug it is, like eventually, or they didn't like develop something like that yet?
Well, so that's what we did, the DNA metabarcoding. So what Ryan did is he dissects out the gut of a mantis, he grinds it up, and he does, this is beyond my expertise, the molecular side of this, but does he has like DNA tissue kits to sort of amplify the DNA, and we have primers, and then sends it off for sequencing. So that's sort of the best we could get, but it does one cost a lot of money. And two, take time. So that's what we're trying to get at with that. I know there have been people that have tried to identify what they've eaten by doing a gut content, but that's also really time consuming, and you have to really know your insects.
Oh, okay.
Right.
I can stop the screen if so I could see everybody, but
There we go. Okay. Um, basic survival strategies. You weren't really looking at that aspect so much, but with all the variation in camouflage, there's a great number of species worldwide that um, inhabit some pretty specific niches, and their coloration and um, ornamentation, I guess you could say, right, is contributes heavily to their success in survival. But yet you're looking at predation specifically in this case.
yeah, so you're right that the camouflage could have evolved for them not to be prey,
Hmm.
but it might also benefit them being a predator. Yes. And we haven't looked at, I've thought about looking at survival locally, but haven't done that yet. But you're right, it could have evolved for not predation, but to not become prey to other predators.
Hmm. Definitely, yeah. Have you worked with any of the South American mantids that they look like they're very spiky and uh, they look like those moss katydids or some that look like that.
I have not. I know what you're talking about. No, I have not worked on those. I'll say yet, because I'm always wanting to do more.
Yep. Good. That's good. All right. Well, anybody else, feel free to ask a question. Um, and the other question, I wonder if the lab that you run, is that something where a student could visit and come and see some of the stuff you're doing? Or is that possible, or is it, you know?
Yeah, potentially. So we're in our new building, which is much nicer than the old building. We have much better animal rooms. So we have a vivarium that is a secure facility, but I we can do tours to show that. We also have some species sometimes on display, what are called our living windows. So some of our classrooms face the inside of the building, and there's tours that go by. And there's one professor that does a lot of science education of arthropods. So we breed some of the mantises when we have surplus. We provide them to her to display on the window as well, just to show some of the diversity. So it's a nice sort of outreach to show them as well. But if you have students who are interested, we could give a tour and show, the lab setup. We're always trying to keep some mantises in the lab for projects. I have some students working on projects currently not right now, but this semester working in the lab. Yeah.
Very cool. That's good. So you guys hear that, students? Could go and see. Um, Now, John LaPola had a insect museum sort of thing there, didn't he? How about that? Is that in the new building now?
We have a much nicer facility for that. Yeah. That's
Good.
also, it's kind of next to the vivarium downstairs. Yes, they got good funding to really upgrade our facilities for the museum collections. Yeah. So we have the herbarium, the insect collection, the wet collections of the fish and herps, and then the dry collection of insects, some mammals, and some birds. Yeah, that's also a thing could be visited.
Cool. Very nice. Very nice. All right. Does anybody else have something they'd like to ask? Anyone? Dr. Mike, you usually have a question.
Fred, I often do, except someone has turned off all of the our faces. You can't see us.
You can put your camera back on. Look at the bottom of your
I
screen.
tried and it said you cannot start your video because it is disabled by the host.
Wait a minute. Hold on a second.
You disabled it. Ah, they
Okay.
just came back on, a
There you
bunch
go.
of others.
All right.
Still won't let me put mine on.
You sure?
I just
Oh, wait a
tried
minute.
again.
Hold on, Mike. Wait a minute.
But Janet's now back on.
Okay, try now, Mike.
Lo and behold, I'm
It's
back.
Mike.
Hi there.
Hi.
Couldn't ask the question until the video came on, though. You do realize, right?
Your whole talk on praying mantis was very interesting, but it's not part that I'm interested in. One thing that Fred sort of mentioned at the very beginning was on, particularly for insects and for other animals, that if they're dying, they will oviposit whatever eggs they have on the possibility that they might survive. Now, one of the things that praying mantises are known for is that they often eat their mates. Why is that beneficial for the male to be eaten?
I mean, what's interesting about mantises is they have asymmetric genitalia, and so they don't always eat the mates.
Right.
When
But
we're
why
trying to
is it
breed in the lab,
beneficial for the male to allow himself to be eaten by the female he just mated with?
I think from a male's perspective, as long as he gets his genes on to the next generation and fertilizes eggs. I mean, you see this in the animal kingdom. I think of like anglerfish. If you know anglerfish, the males are really tiny and basically just become spermatophores and attached to the female. And so it's for
Right.
them, it's I think a pass
Well,
along my gene.
the reason I've always thought for years is that if you have fertilized a female mantid, she needs a certain amount of food to develop those eggs. If he mates with her and leaves and she doesn't find enough food, his genes are dead.
Yeah.
If he his
Yeah.
genes go forward.
I
Therefore, it is worth sacrificing your life so your kids will live.
I agree with that. That's a good point. I never thought about it necessarily from that perspective, but
I
I
think of
mean,
things differently.
I mean, it's pretty amazing because we will see them rip the head off the males and the males still be attached and on the female, even without
Well,
a head.
the similar one is spider. A lot of spiders, where when they mate, the female will then eat the male. There
Hmm.
are other spiders that have learned that if they bring their female food, while she's eating the food, he copulates and gets away some of the time. Other ones have learned that if you wrap the food in silk and give it to the female, by the time the female can get the silk unwrapped and eaten, he's escaped.
Huh.
Then other males have learned, what a waste of time. Let me give her a bag of silk. By the time she realizes there's no food in there, I'm gone.
Huh.
Mhm.
So it's all, again, different way things have evolved
Yeah.
and strategies.
Yeah,
So
that's cool. I didn't know the
it's
spiders.
unrelated to your talk, but just fun stuff.
Yeah, no, that's cool.
I wonder if the male and the mantises again, when the male is getting his head ripped off and all that, and they're attached and they're copulating at this point. Is there any um, pulsating or any kind of muscular movement on the males that enhances the copulation and in the throes of death, if you will, right? Does that have any effect on the copulation?
I'm trying. I want to say yes. I feel like I've seen this in the lab, but
I
we've
think I
not.
heard this before. That's why I asked.
Yeah, I mean, some species, and there's also dimorphism among the species too, with males being smaller in some. But I'm pretty sure we've seen that in the lab, yes, where there is sort of still muscular movement to make sure, right, the genes are getting
Yeah.
passed on.
That's what I thought I had heard once, but I wasn't 100% sure. But anyway,
I have not heard that,
hmm.
but it's not something I would have studied. My work is with mosquitoes and viruses.
I mean, I'll pay more attention to it. I mean, the mating is interesting. It's, we try to mate sometimes just to have individuals to work on, and it's always we have to be careful depending upon how many males. We have to make sure the female's well fed, while the males, sometimes the males are on there for a while, sometimes they're not. It's the mating behavior variation is really interesting among the different species as well. And um, we're always sort of keeping an eye out for that stuff. But sometimes you leave a male and female, and the male gets eaten, and you hope his genes got passed on.
Yeah. Okay. Well, I'm looking at the list here and see who else. Does anyone else have a question that they want to ask? Um, any of our members who are present that care to ask a question? Anyone? If not, it's okay.
I had a quick question.
Sure.
Um, I noticed that there's, like you mentioned, there's a pretty extreme morphology for some of the mantis species, and you mentioned rockfish as another species that has some pretty significant changes for morphology for camouflage. Like looking at apex predators, not that they don't have any camouflage, but it does seem less extreme, I guess you could say. Does that maybe point to the possibility that it's being both predator and prey leads to more camouflage, or not really?
That's a great question and interesting. And yes, I would say potentially because mantises are also preyed upon. But even in some, I always think of, and I use this in my animal phys class, like sharks and other apex predators, they do have that counter gradient coloration, which benefits predation too. And I think what's hard about camouflage is how we perceive things is completely differently than how other things perceive things. So we're very anthropomorphic. But I do think because they are smaller praying mantises and they're not the apex top of the food chain, they are trying to hide from whatever's trying to eat them, like you said, but it could also be benefiting them in their predation as well.
Thanks.
Well, we don't want to keep Dr. Oufiero too long. It was so nice of you to agree to do this lecture tonight, and we thank you again. On behalf of the Maryland Entomological Society, we do have some things we'd like to send you. So we have a year's membership and a journal and something else as well. So if when you get a chance, could you kindly forward a mailing address that you prefer that we send you these things, okay?
Yeah, can do
And,
that.
and we'll get it out to you. All right.
Thank you. Well, thanks for having me and listening to about what our lab does.
Absolutely. Thank you for sharing. And it's really nice to hear about this. And um, yet another great thing to know about Towson.